So now we can tackle our third major class of macromolecules. We've looked at carbohydrates and we've looked at nucleic acids. So now let's look at proteins before finally moving into lipids. So we've already talked about protein functions. Proteins can serve a variety of functions, and proteins are polymers. These are macromolecules after all. And the monomers that are polymerized into proteins are amino acids. We'll talk about that in a second. So a protein is one or more long unbranched chains. Some proteins are a single chain of amino acids linked together. Other proteins are not functional unless several chains of amino acids linked together come and bind to each other, and then we have a functional protein. So we call each chain, each polypeptide chain, which is a polymer of amino acids, we call that a polypeptide. Amino acids are the monomers that are linked together to form proteins, and we can talk about amino acid structure briefly. Every amino acid has the same basic structure except for a group of atoms that we'll call R, and R is variable. But every amino acid has an amino terminus, an amino group, a central carbon that we'll call the alpha carbon. That alpha carbon has a hydrogen, is covalently bonded to a hydrogen, and is also covalently bound to an R group that distinguishes a particular amino acid that makes it unique among the amino acids. So the R groups impart the particular chemistry to a particular amino acid. We'll talk about more about those in a minute. And then finally, we go from amino alpha carbon to carboxycarbon. Here's a carboxyl group. So amino on one end and a carboxy group, which is acidic because hydrogens can be donated to solution. So amino acids. And the R groups are the parts that are variable. Oops. So how do individual amino acid monomers come together to form a protein chain, to form a dimer and then a trimer and then a polymer? It's a condensation reaction. Water is removed and the carboxycarbon of one amino acid becomes covalently linked to the amino nitrogen of another amino acid. And hydrogen, oxygen is released in the form of water, molecular water. This is a dehydration or a condensation reaction. And now we have, in this case, we have two amino acids joined together, so we have a dipeptide. This is the peptide bond between the carboxycarbon of one amino acid and the amino nitrogen of another. That is the peptide bond. And it has special characteristics because the electrons found in the double bond between the carboxycarbon and this oxygen now, those electrons can actually resonate and come over here and impart a partial double bond character to the peptide bond. Here are the R groups of two adjacent amino acids in a dipeptide, and they, as you can see, they are sprouted off the alpha carbon. Here's another way to look at it: amino group, carboxyl group, and alpha carbon, and the side chain, R. And in a long chain of amino acids linked together, it is the peptide bond that links individual amino acids to each other. Now, before moving on to 4 levels of protein structure, for the hierarchies of structure that determine the final shape of a protein, I'd like to talk a little bit more about the amino acids because I think it's fairly important at this point. Now we can study the various amino acids in terms of their R groups, which are the only invariant thing, the only thing that differs between the amino acids. So every amino acid has an amino group. There's a nitrogen with hydrogens attached. Hydrogens are white, hydrogens are in white, nitrogens are in blue, carbons are in gray, oxygens are in red, and the only carbon that is not gray is the alpha carbon, colored in green. So every amino acid has the same basic backbone structure, and these are called the main chain atoms. So we have nitrogen attached to the alpha carbon, attached to the carboxycarbon. And carboxycarbon has 2 oxygens attached, is bonded to 2 oxygens. Now, we can group, the only thing that differs is the R group that is attached to the alpha carbon. That's the only thing that's different from amino acid to amino acid. Yellow, by the way, is sulfur. So there are 2 amino acids that have sulfur in them, methionine and cysteine. And we'll see this sulfhydryl group here is very important in certain cases because 2 sulfhydryl groups can come together and form a covalent bond between the S's, and that can stabilize protein structure. It would be like putting a cement reinforcement across a bridge, holding up the spans of a bridge together. So cysteine is very important in that regard. Let's look at some of these R groups and characterize them in terms of their chemical properties. Alanine has basically a methyl group, CH3, on it. Valine has slightly more complex hydrocarbon group on it. Leucine, carbon, carbon, carbon. Here's a methyl group, and CH2. Isoleucine. And what do you notice about these so far? You notice that these R groups are all hydrocarbons, meaning they are non-polar. And in fact, we're coloring the non-polar amino acids, we're coloring their names gray to designate that these are non-polar amino acids. So we have all the way up through glycine, which essentially only has a hydrogen as an R group. All the amino acids have 1 hydrogen attached to the alpha carbon, and then the R group is bonded to the alpha carbon as well. Glycine has a hydrogen, but then its entire R group is just another hydrogen. So glycine is the smallest amino acid, and for that reason, when glycine has imparts great flexibility to a protein chain. Now, proline is interesting because its R group actually cyclizes and covalently bonds to the amino nitrogen. So proline actually can form a kink in the chain of a protein because it folds back and its R group is covalently bonded to the main chain of the protein. Now we move into polar amino acids that are not fully charged. There's some polarity in the R groups, meaning they like water. Serine has an OH group on it, and that we know that this is a polar covalent bond, and so O will have can have a partial negative charge on it. The same with threonine. There's an OH group, a hydroxyl group on the R group of threonine rather. Glutamine has an amino group on it as well as an oxygen. Asparagine has amino group as well as an oxygen as well. The only difference between asparagine and glutamine being that the chain is longer in glutamine. The hydrocarbon chain is longer in glutamine. Cysteine, CH2, and then a sulfhydryl group. That sulfhydryl is polar. Tyrosine has a hydroxyl group at the tip of this aromatic ring as its R group. Now we can move into the acidic amino acids. The acidic amino acids and the basic amino acid shown in blue here, designated in blue, are capable of having a full charge on them, whereas the polar amino acids, serine, threonine, glutamine, asparagine, cysteine, and tyrosine, are polar, but don't have a full charge on them. They have partial charges on some atoms in their side groups. But the acidic amino acids donate hydrogens to solution because of their R group oxygens capable of donating hydrogens, and they therefore, when they donate hydrogens, they become negatively charged. So acidic amino acids can have a negative charge on them, a full negative charge, when they donate hydrogens to solution. Whereas the basic amino acids can be positively charged because their amino groups on their R groups can accept hydrogens from solution, thereby lowering the hydrogen ion concentration. And when these amino groups accept a hydrogen from solution, they become positively charged. So basic amino acids are positively charged, or capable of being positively charged, depending on the pH. And acidic amino acids are negatively charged, are capable of being negatively charged. Basic amino acids, positive. Acidic amino acids, negative. Now we can move on to the 4 levels of protein structure. And this is very important because it is, in the case of protein function, function follows form. The shape of a protein will determine its function. The shape of a protein can create a binding pocket that is very specific for molecules that the protein might interact with. The shape of a protein can bring together in an active site amino acid side chains of the protein that can catalyze a particular biochemical reaction. So in the case of protein structure, function follows form, and we therefore need to understand how protein form is acquired. And in these 4 levels of structure, we start out with primary structure. And a primary structure is simply the sequence of amino acids in the protein chain. So if we were to just do primary structure here, it would just be the amino acid 1 linked to amino acid 2 linked to amino acid 3, and so on to amino acid N, whatever that is. The sequence of amino acids defines the primary structure. Secondary structure is formed by the interaction of groups in the backbone of a peptide. A peptide is 2 or more amino acids linked together. And if we consider only the main chain atoms of a protein, you might have R groups hanging off the amino acids. R groups would be hanging off. But if we forget about the R groups temporarily, just forget about those, and just imagine the main chain atoms of a protein. That forms a long chain, and that chain can fold into particular shapes that define secondary structure. And they are of 2 types, alpha helix and beta sheet, and then the loops that connect alpha helices and beta sheet. So an alpha helix is when we coil the main chain atoms of a peptide. And there are interactions, hydrogen bonds in fact, that occur between main chain atoms about 3.6 amino acids apart and stabilize this helical structure. That is the alpha helix. Beta sheets form when part of a protein, call that a strand, a beta strand, can line up with another stretch of the same protein, or a different protein, but usually it's the same protein. And hydrogen bonds can form between main chain carboxyl oxygens and amino nitrogens. Hydrogen bonds can form there. And if you can have several of these strands, more than 2 strands, participate sometimes. And when that happens, you end up with something that we call the beta sheet. And it's planar. If you were to look at it from its side, it might look something like this. It's like a plane. Now, we'll talk about a few examples of the beta sheet and alpha helix shortly in our next segment. And that's where we'll pick up with for the final part of this particular lecture.